Hello, it is a great pleasure to have this opportunity to introduce the project SoilLife1st. I am Maria do Céu Godinho, I work at Escola Superior de Santarém, and I'm a professor of crop protection. SoilLife1st is the continuity of a work that we are doing since 2017 to promote soil health in agricultural systems with strong crop intensification, high levels of fertilizers, water and pesticides. The enormous problems related to the resources management and conservation, as well as the need to protect crops against pests and diseases, imposes the need to carry out a path that is long, but is already causing an impact. The main objectives are to improve soil fertility, increase organic matter and water retention and storage, and promote biodiversity. This aspect is crucial when we think about its functionality. Huge functionality. Soil biota, mesofauna and macrofauna are key elements in this process. It is these aspects that we'll present here as a current case in a living lab format, structured and supported by SoilLife1st project. I will recover the introduction we made to the SoilLife1st project and remember that one of the motivation is the resolution of problems associated with pests and diseases, particularly of intensive crops such as processing tomato, potato or maize. On the one hand, the need to reduce the use of pesticides, and on the other hand, we are in a period of banning some undesirable substances from the market for their side effects. Both aspects lead to the effective need to reduce pesticide dependency. This is fully in line with the Sustainable Use of Pesticides Directive as part of the European Green Deal and Biodiversity Strategy. The core theme of this module is to highlight the importance of biodiversity and its functionality related to crop protection, the monitoring and the effect of cover crops evaluation. In the first place, the most modern solutions for the protection of plants, in my opinion, should include integrate technology, digitalization and precision, no doubt, but always with the support in the principles of agroecology. It is in ecosystem services that we must support the dynamics of the processes. These begin in the ground, in the soil. These begin in the soil. Knowledge has evolved a lot in recent years in this regard. It is known the role and involvement of many organisms that habit the rhizosphere in plant nutrition, but also in plant defenses process. Is much more what is not known yet. But what is known is very promising to convert us to this agroecological transition that is urgent. So what are we doing and why measure biodiversity in the soil? Because it's soil life that ensures functions like the composition of organic matter, conversion, mobilization, and some more questions. Because arthropods facilitate the degradation of organic matter, because atmospheric nitrogen is fixed by root nodule bacteria and provided to host plants, because there is improved absorption of nutrients by associations between plants and fungi, like mycorrhiza, because many other species present as PGPR are very important to defense mechanisms. In the SoilLife1st project, the challenge is to introduce cover crops in each farmer's technical profiles and evaluate the effect on soil biodiversity. Fertiprado, a Portuguese seed production company, prepares mixtures adapted to the realities, soil, ecology, and possible period between the main crop in each agriculture year. Regarding biodiversity, we are developing a way to communicate with indicators of easier perception. A good example is KBS. It relates the presence of certain species more adapted to the soil and its quality. For now, we are satisfied with the response and we are continuing to improve it. Working in a network with Italian colleagues who developed this method, and as we can see from the images, it is important to find a way to communicate soil evolution based on the biodiversity of microarthropods measured before and after the introduction of cover crops. As you can see, the KBS value increases in the soil samples after the cover crop introduction. It's important to inform as well that we are also evaluating some other groups of species like carabids, earthworms, and others. The microbiome, which is very important, such as bacteria, fungi, nematodes, is a work done by INIAV team, which is a very important partner in our project.